Okay, so welcome everyone, and thank you again for joining us for another session of the PCE3 seminar series. And I'm sure most of you know that PCE3 stands for Prebiotic Chemistry and Early Earth Environments. And one of the main goals of PCE3 is to bring together researchers with different expertise to collectively work towards a better understanding the origin of life. So today, we're excited to have Dr. Indrani Mukherjee, who is a lecturer and researcher in earth science at the University of New South Wales. And Dr. Mukherjee earned her PhD at the University of Tasmania. And afterwards, she stayed to work there as a lecturer and a postdoc before accepting the Roger E. Dean Fellowship at the University of Toronto. And her work is focused on ancient marine environments and explores the links between early Earth evolution, the origin of life, the origin of complex life, and the formation of precious mineral deposits. So thank you again for joining us, Dr. Mukherjee. And whenever you're ready, you can go ahead and share your work.
James, can I ask if you can see my screen?
Yeah, I can see your screen. I think everything's working. Yep.
Thank you. Well, thanks everybody for joining this seminar series. I'm really grateful for the opportunity. I would like everyone to know that this is I'm usually working on ancient marine environments and how ocean chemistry changed through time and how that's impacted evolution of life. But in the process of gotten interested in biogenicity and sort of sometimes dabble across in the field of astrobiology. But in no way am I a bona fide astrobiologist. I think you can tell, you will be able to tell from my presentations. But anyway, so today I want to talk about the fingerprints of ancient life on Earth. But before I begin, I'd like to acknowledge that the bulk of the data that I will be showing you today was collected on the lands of the Muwinina people, my previous employment location, University of Tasmania. And the Muwinina people are one of the oldest continuing cultures in the world. And I pay my respects to our elders here in Australia. So I'll dive straight into it. I've usually worked on the trace element chemistry of pyrite in marine sediments for all sorts of things. So we use the trace element chemistry information for mineral exploration studies. We use the trace element chemistry to understand how the ocean nutrient conditions change through time, and also to infer past atmospheric ocean conditions. So we've been working on growing this large body of data for the last 10 years. And in the process, while we analyze our pyrites using in situ techniques, we have this textural context, which has been very important for us. For instance, we want to analyze sedimentary pyrite and exclude the hydrothermal, depending on whatever the research problem is. So in the process, I realized that I was coming across a lot of textures that looked biogenic. And I would always put it in my manuscripts or papers and always have those textures as biogenic with a question mark in brackets, because there was no way of saying or being certain that it was in fact biogenic. Just because it resembles biogenic textures does not mean anything, because we have things like biomorphs. So I was always curious to basically understand if there was a way of distinguishing between what's biological and what's non-biological. Especially in my case, where I'm looking at pyritic textures, it's obvious that whatever the organism was is obviously replaced by pyrite. Now, we know pyrite is a mineral, and minerals are ordered structures, but biological organisms are also ordered, but I think it's more organized, as they say. So is there a way where we can use the chemistry of pyrite to distinguish between mineralogical ordering versus biological organization? Now, this is a very big question, and I want to say upfront that I haven't found the answer, so please don't be excited. But that's the main question, really. And you'll see, just starting to scratch the surface of it. But I want to just go through some basics first, just to show you how deep the problem is. So biology, as we know, can induce mineralization. They precipitate minerals for all sorts of things. But there are two main kinds, and they are either biologically controlled or either biologically induced. They can form the exact same minerals. So in this case, I'm showing you, let's see if I can find the laser so I can point easily. So in both cases here, it's magnetite that's being precipitated, but one is biologically controlled, where the mineral is actually precipitated within the confines of the organism, and it actually serves some physiological function. I think magnetotactic bacteria uses that for location and chemotaxis, etc. But biologically induced mineralization, in this case, acicular magnetite, is essentially the same mineral, but in this case, the magnetite is formed on the outside, beyond the confines of the organism, and it's a product of interaction of the organism's metabolic waste with the surroundings. So there are these two types, and I don't want to get bogged down with what's controlled and what's induced. At this stage, I'm just very keen to find out if any of the pyrite textures or silicate or calcite textures that I come across have any biological affinities to them. And then the other two things that we have to remember is the difference between permineralization and orthogenic mineralization. For a long time, I thought what I was looking at was, permineralized pyritized fossils, etc. But there's a, very stark difference between the two. So permineralization is basically very slow and fine and precise replacement of a cell by cell of an organism by a mineral charged water. In this case, I'm giving you an example of the cell wall tissue structures that have been silicified. And then on the other hand, we've got permineralization, which is a product of the irony, decay. And you might think decay is bad if something has to be preserved, but actually it's not. It's the decay has to be, the decay rate has to be lower than the basically the formation of the other minerals. The decay cannot exceed the formation of precipitation of the minerals. So in this case, the decay actually helps with the mineral precipitation, and that mineral then replaces whatever it is that's been decaying. So there are two main differences here. So one is early infiltration and permeation of tissues, and the other is a product of activity of decay bacteria. So they're two different things. So I'm more on the orthogenic mineralization side of things, since I'm looking at pyritic textures in my rock. So this is a beautiful preserved wood fossil from Canada. This was a sample that I was studying for a totally different reason. I was looking at the why these shales are, metalliferous, and what critical elements can we find in them. And, you bump across these beautiful textures and you think you've got to do something about it. So just trying to understand why these textures form on the first place. It obviously depends on the amount of decay. And then obviously you want to know what the nature of the microbial activity is that's causing the decay on the first place. And of course, you want to have the mineral ions present. So in my case, I will need iron and sulfur, obviously, if my organism is going to be replaced by pyrite. And sometimes, so the precipitation is obviously microbially induced, but sometimes the microbes itself, not in this case, of course, the photograph that I'm showing you here, but sometimes the microbes itself just get also lithified, and it happens with apatite a lot. And then, of course, the size of the crystals. So the smaller your mineral crystals can form, the finer the details you preserve. So in my case, pyrite is good for replacing the mineral, but it's not as good as apatite, because apatite crystals are much finer. So the level of detail preserved is much enhanced. And then, of course, we've got taxonomic controls on the organism. You know, what part of the body is being preserved? Is the soft part? Is it the hard part? Is it the muscle part? And there's all sorts of things that needs to be taken into consideration for this. So I was just having a look at this fantastic paper, and you know, I'm not a chemist, but it's a really good one to get your head around. Basically, all the processes that controls the process of crystallization, and it's incredible. This paper actually talks about how they are basically creating bio-inspired materials, in the sense that they're understanding how biology creates new minerals or mineral morphologies, or controls mineral morphologies, and how that information can be used to synthesize new materials in the lab. So I was just trying to draw some inspiration and see what they know about how biology actually works, to see if that can help me interpret my data well. And turns out there's all sorts of things, and I've just given you 3 examples here. So the availability of macromolecules can have a strong influence on mineral morphologies. What kind of polymorph is present? So I know for a fact that we've got pyrite and marcasite. Is there a preference? Because we know that organisms prefer certain polymorphs for their own physiological function. And then of course, crystal orientation. I'm giving you an example of the coccoliths here, where the certain crystal, they prefer certain crystal faces for growth. So there are all sorts of things, and this is just, very few. And I've just started delving into this as well. But it's very interesting to understand how biology or organisms have developed strategies to basically precipitate minerals in the way that's favorable to them. So that's good to know that there's this really strong connection. But that's not something that we haven't known. We know that they have a huge influence on the crystallization process of forming amorphous minerals, etc. It's how can we then detect it in our rocks that's really, really old. We can do these exercises in the labs and collect all this information, but how does that then translate into what I come across, for instance, in my rocks when it comes to pyritic textures? And here on the left, I'm giving you an example of some of the textures that I come across. And I think it looks very biogenic, but what I think makes no difference to anybody, because I don't have a way of ascertaining yet that these were in fact biological nature. And the other problem as we go further back in time is the size. So as the size, as we know, as we go back in time, the size of the organisms, tends to decrease a lot, and they're all microscopic. And that makes it even harder. They're small, but they're very significant. And so with, advent of new techniques such as a synchrotron, I suppose we could get into the middle of all of this. So detecting is never easy. However, if it's preserved very, very well, in this case, I'm giving you an example of preserved fossils from Bengtson et al. Paper in 2017. It's pretty straightforward. Here are some exceptional preservation, subcellular, would you believe, of a giant ostracod sperm in Australia. That's pretty incredible as well, if you ask me. And sometimes, and thank God, because I love the fact that they've been pyritized, it can be pyritized. And my aim is to basically find out if there's any way of looking at the trace element chemistry of pyrites in these preserved, orthogenically mineralized samples, to see if we can find some signs of biogenicity. But also note in this that the replacement occurs by pyrite with different habits. So the first one, you've got basically euhedral cubic pyrite replacing the ostracod. In this case, you're forming framboids, and that's very interesting to me, the morphology side of things. And it's essentially the calcite replacement in pyrite. That's been studied a lot in economic geology, but it's also very important in terms of calcite replacement. in pyrite. The other important thing I should mention here is that these replacements or autogenic mineralization occur in these microenvironments. That's why I think that the trace element chemistry of the pyrite that's forming in these microenvironments would be different to the ones that just generally forming in the sediment. So for this replacement to occur from calcite to pyrite, this occurs within the confines of the decaying organism, and it's highly controlled. So the iron and the sulfur, the supply has to be rather slow and steady. If it's too much, then you'll just end up precipitating tons of massive pyrite. You probably won't preserve much. And you just have this, rim of pyrite around this decay organism. But when it does tend to get replaced nicely, it's a very controlled environment. And I sometimes feel that perhaps that controlled environment will have a different trace element signature compared to the rest of the pyrite. Similarly, for plant fossils, they're also quite commonly replaced by pyrite, and a lot of experiments have been done on the process. So here's how the different morphologies of pyrites can replace the plant. So it can be anything between framboidal pyrite to framboidal, as well as euhedral pyrite, or very coarse polycrystalline pyrite. And it all depends on basically the EH/pH conditions. Of course, the chemical controls that normally are important for the precipitation of pyrite still remain. So here's a beautiful example of exactly the same thing. This is from obviously Eocene London clay, but I was looking at my rocks from Canada, which is the Devonian, I think. And they also have these beautifully preserved wood fossils. Here's another example. I think I've shown this slide to you before, but it's very, spectacular. And I've been thinking of what can be done. Here are some more textures that I'm going to show you in the next few slides that I come across to see, well, what does this mean? Here are some samples from the MacArthur Basin, and this is very close to the famous, here's a chance or MacArthur River zinc lead deposit. And it was funny because we were looking at this from a mineral exploration point of view to use the trace element of chemistry of pyrite for vectoring and fertility. But we came across these textures, and these textures were only present close to the ore body or the mineral deposit, not further away. So that was interesting. And these filamentous looking things. And then you start to play with your imagination. And by the way, I'm not saying it is paramecium. It looks like paramecium, right? So I was like, wow, that's cool. Is it though? And similarly, you come across these textures and you're like, could it be this? Could it be that? And they were all from this close to the mineral deposit side of things. And then you can look at it, the SEM, and, go wild with your imagination, basically. But just because it looks like that, does it mean anything? So I'm going to give you 3 examples here. And, at first glance, if you look at it, you probably think, oh, well, yeah, they look pretty similar. But the first one is a photomicrograph of heavy minerals in a thin section of a tungsten. Got nothing to do with biology. The second one is to do with biology, and I will not try and pronounce the name. I'm not a biologist, and even if I tried, I think I'm do a terrible job. And the third one is rosettes of tubes emerging from seed crystals. This is actually a really fantastic paper by McMahon 2021, looks into the various different kinds of biomorphs, or he calls it, or they call it the chemical gardens. And they plant light structures, and they can just be, formed synthetically in the lab. There's no biological affinity whatsoever. So we can't just go by looks. There's no way we can say that just because it looks like that, it could be biogenic. You've got to have a question mark on that. But there are indirect evidence. So we published a paper and looked at nitrogen concentrations in the shales close to that old deposit that I was talking to you about, that I worked on, and further away. And we saw that there was more bioavailable nitrogen close to these venting sites and further away. And so we can make these indirect references and say, well, since bioavailable nitrogen was available to these organisms, so it is possible that these are biogenic textures, since nitrogen would have been required. So there are these indirect references that we can make, but there's no sure way of saying. So I thought of like I said, the whole premise of this was to look at trace element chemistry. But just to show that I'm not the first one to do this or anything, people have looked at a concentration of elements in preserved fossils or textures to see if that can be used. And usually carbon is a very good way of, or has been proposed to be a good indicator of biogenicity. This is a really good paper by Boyce et al. And it's a bit of an inspiration for me really to understand how and why certain elements are concentrated in certain parts of the fossil, and if that can be used to our benefit. But the motivation of using trace elements in pyrite really came from this paper, Marshall et al., 2017, where they used synchrotron techniques to detect the presence of vanadium in microfossils, and that was their in their microstructures or whatever they thought looked biogenic. So the concentration of vanadium ended up being a significant tool for biogenicity. And I thought, well, we also analyze trace elements in potential microstructures all the time. So could we use some of that information for the purpose of biogenicity? And so that was the idea, but this was the motivation. And funnily enough, I remember applying for a small grant at the university and saying, instead of using the synchrotron, I would like to use a more readily available technique, which is the laser RCPMS, which was readily available to me. But I've come to the conclusion that I have to use the synchrotron now. I'll explain why. But here are my samples, just the ones that I wanted to try on. So we've got some non-pyritized as well as pyritized ammonites. And so we wanted to do some high resolution microscopy to see what the textures are like. And if we can distinguish between what could be potentially biogenic and what's not, could we then get some more textural context, crystal structure, crystal orientation? Could we then add compositional context to it and come up with, I don't know, fancy statistical techniques? These days, AI can do pretty much everything. But we used a very basic one, but this work is ongoing, so any advice would be really welcome. So we use, clustering as well. So a combination of textual, compositional, and statistical analysis to see if we can come up with a tool for biogenicity. Just very quickly, what elements? I know we focus on oxygen all the time. I love oxygen, that's fine, but I think trace elements are also very important because there's a fantastic paper by Kobayashi and Ponna Peruma. It just goes to show how important trace elements have been in chemical evolution and how organisms have evolved to preferentially incorporate trace elements for their function or not. So it's really important to understand trace elements, in my opinion, because they have a huge role to play. And we all know what trace elements do for you and me and organisms, but really, it's like I said, it's the preferential incorporation of certain trace elements over others by biology that probably makes trace elements really important, because the major elements, the CH, NOPS, everybody requires that. Everyone's going to need it for all sorts of things. It's the building blocks of life and all that. But trace elements can be that preferential requirement that might make them interesting candidates for biogenicity studies. And it is, because this is, again, I'm not a doctor, please, but I bumped across this paper. People, even today in medical science, they're using the concentration of elements in organs and tissues to delineate the different structures within the cell or the tissues. So it's only normal for us to think that trace element concentrations in organic materials would give us a useful insights into these things. So just quickly coming back to my samples, here's the outcrop where the samples were collected from. They were collected by my supervisor. Unfortunately, I wasn't able to collect it myself. I wish I would one day. So backscattered electron images, just to have a look at any textures that's preserved, because ultimately ammonites are hollow, and if all I get is the precipitated, even though it's a microenvironment of its own, but I'll probably be just getting the chemical precipitation in a hollow space in an ammonite, which is probably not bad. But I was thinking perhaps there's some feature in this that might be preserved, which might have been primary. So if you zoom into that red triangle here, you see, I'm just zooming in, and you start seeing some features that start to tell you that perhaps this was some material, there was an organic template. And, while pyrite precipitation is going on the hollow parts, which is rather coarse, infilled by further the pyrite. By the way, all of this is pyrite. We've analyzed it and it's all pyrite. And then you see these beautiful textures that have also been preserved by pyrite. Some more textures that we see preserved. And so once you start seeing this, and this is the calcite, the non-pyritized versions of it, here also you start seeing some differences in the way, it's the same mineral, right? But it's because I think it's different parts of the fossil being preserved, perhaps soft, or a different part of the muscle, or it's just taxonomic controls, as I said. I think that the kind of mineral that dictates the kind of precipitation, and that's why you end up getting these different textures. So you can do some crystal orientation maps here, which is what I've done, and they seem to be very different in the different worlds, as they say, in the ammonite. But I wanted to get the compositional context. Once I had this textural context, I wanted to see, well, if that's more biogenic, that's not. Well, let's see what the composition tells us. I know this looks like a black box, but this is an incredibly useful instrument. I've used it a lot for all sorts of things. It's the laser ablation machine. And I know it looks like a black box, but a lot of cool things happen inside it. So we did some laser analysis. And we started to see some difference in the fossil pyrite versus, pyrite that's in the matrix in the shale. And there are significant differences. But that could be because of anything. Perhaps the ones on the outside formed a bit later. That might have been something to do with available. I don't think so, because the textures look very similar and sedimentary or syngenetic. But still, there is a difference, which gives me hope. Similarly, we did some rare earth analysis of the carbonate side of things, and that also gave us some differences. But that's not enough. I think what we then did was to image the whole sample. So in this case, we've imaged this portion here completely for the various elements. So here I'm showing you a map of this area here. They've all been analyzed for these various elements that you can see. So you can see there's been enrichment in molybdenum, for instance, on the outside, and this inner bit here that I was talking to you about earlier, which looked very biogenic. And this is bona fide fossils, right? So these are apatite sternkines from the small shelly fauna in the Cambrian, in the Georgina Basin in Australia. And we know these are fossils, as they've been proved. And usually people focus on elements like phosphorus. Oh, it's full of phosphorus. Look at this, full of phosphorus. That's the indicator. But that's the danger of using one element. In my case, if I'm looking at pyrites, phosphorus doesn't actually go into pyrite. So it's the same fossil, same structure from the same formation, not calcite, obviously. This is a pyritic one. You don't see the phosphorus. So now what are you going to say, that it's not a fossil? Phosphorus doesn't go into pyrite. So that's why we're not saying it. And so my point is we should be looking at several elements and see. And I think once you start seeing these different variations within this body, so, sulfur and iron-wise, they look pretty full. But when it comes to trace elements, you start to see these contrasts between within them. And that, I don't know, makes, again, I'm hypothesizing here, so please feel free to critique me later on. But it's perhaps to do with the different parts of the organism that's either soft or hard or intermediate that sort of dictates how much trace element goes into them. Because we know finer iron sulfides, because of higher surface area, they tend to absorb more trace elements than the coarser ones. The coarser ones actually tend to absorb a lot of them. So this could play a control, but let's just keep going. So we've analyzed the sample for geochemistry. So I showed an example here. Just quickly go back. So this is the laser image, and if we go back. So what we did was assign areas. So we thought A was, parts of the fossil remains. B is the interior of the volume fossil. and C is obviously the exterior. And this is where statisticians can do all sorts of things. Sometimes, honestly, I don't understand what's going on, but it works, obviously, beyond my human comprehension, I tell you what. But essentially, what I've been told is, my collaborator tells me, that we take the data from these regions and then we look for clustering. We try and see what the clustering tool tells us to see. So this tool has been used to basically look at how forested an area is. So the density of trees, are they closely spaced, or not? So this is the same sort of phenomenon. We want to see how clustered certain elements or the spatial distribution of certain trace elements are across the biogenic areas versus the non-biogenic areas. And so when it's significantly above the 95% confidence interval, then you can confidently say that, you see high levels of clustering. And so when we applied that to a variety of our trace elements, we noticed that our biogenic bits showed consistent clustering. So for instance, in this case, if you look at iron, cobalt, and nickel, we see that the non-biological areas do not show as much clustering as the biological areas. So we've done that on lots and lots of samples, and we almost always see that the biological areas have far more clustering than the non-biological areas. So we tried to do this test sample. We had no idea what was going on. It was pretty, like a grain of pyrite here, which is pretty, iron and sulfur, so pretty solid. But when you actually scan it for the various trace elements, you start to see these weird structures emerge. And so we thought, all right, well, let's give it a go and have a test. And as we expected, this inside bit showed this sort of clustering that the rest of it, which is also pyrite, didn't show. So that could mean something. But there's more things that we need to do. Trace element abundance itself is not enough, I don't think. I think I'm more interested in the sort of the spatial distribution. And like I said earlier, if I can somehow distinguish between the ordering of a mineral versus the organization of an organism. And so for that, we applied for some beam time at Soleil, and we've been successful. So watch the space. We will probably be doing some really high resolution mapping of these fossils. But additionally, we also, since the synchrotron technique actually allows us to do that, also see the difference in the speciation. Perhaps the biological areas will have a different speciation of trace elements compared to the non-biological areas. That's essentially my hope, but we'll see what happens. And of course, sulfur isotopes will also provide additional evidence. So it's a combination, it's a work that's ongoing. And just wanted to show you this paper that's been recently published. And I think it was pretty evident, because there's so many biomorphs and so many structures that can be synthesized in the laboratory without any influence of biology. I think we've come to realize the fact that it's in our best interest to study these environments. So for instance, in my case, perhaps I could look at some modern vent environments and look at how the chemistry works here. So that will probably better help me to interpret what's going on in an ancient vent environment. And yes, lots of statistics. I'm still trying to get my head around it, but there are lots of different ways of essentially, good discriminator diagrams where you can basically tease out populations where it tells you what's a biological influence and what's not. And this is going to be very useful. So we're going to have sample return from Mars in about 10 years' time. So our biggest asset, I'd say, would be our understanding of the various different styles of preservation that we have on Earth. And if it's a fossil, then obviously it's a no-brainer. But if something's been completely remineralized, then do they still preserve some evidence of that it was once a biological organism? And I think this is my last slide. Yes, this is my last slide. And I just wanted to end on this by showing you how little we know. So we want to study how life evolved through time, etc. But we actually don't have a full account of what life looked like in the past. We don't have a full account of what our total biosphere looks like right now. So there's lots more to be explored, which also then tells you that also means that we're probably missing out on a lot of information. In other words, which organisms prefer which elements, etc., and their requirements, and what minerals they cause to form, and what geostrategies do they adopt. So I think this work is ongoing, and the more data we collect, the better or the closer we're going to get to developing a robust biogenicity tool. And I'll end here. Thank you very much.
Great. Thank you very much for sharing your work. And I think everyone's excited to see what you're coming out with in the future. And so now, if anyone had questions for Indrani, feel free to type them into the chat and we can read them out. Or if you prefer, you can raise your hand in the Zoom features and we'll ask you to unmute and you can go ahead and ask your question directly. And I think there were already some questions in the chat.
Do you want me to read the chat?
Sure, I could read them out to you. So I guess some of the other people can hear what the question is also. But if you want to read along also, that would be great. So I think the first question was from Paul Shapshak, and he asked if you'd done any analyses on meteorites, especially from Mars.
No, unfortunately not. But this year we had the astrobiology conference, and thanks to Albert and Anna at UNSW for organizing that, because I got to meet some people and they're based at university here and at Monash, who work on Martian meteorites. So they're quite keen, and they have some pyrites in their samples, and they were like, would you be interested? And I was like, yes. So really excited about that. But no, I haven't analyzed it yet, but I would love to get my hands on some samples. They're very hard to get. What is the dimension of the smallest microorganism you've analyzed? Well, I guess these ones here. So let me see. It'll probably be 20 microns, which we know is not the smallest size of a microbe. I think I would say 20 microns. I'll just keep going back.
So related to that, what is the spot size you can get on the ICP-MS versus the synchrotron?
Yeah, so that's the thing, right? So I started off by saying perhaps we will not need the synchrotron. And so this is probably the smallest, I would say, 20 micron, to answer the previous question real quick. Yeah, 20 microns or maybe 10, but that's about it. And the laser resolution was, the smallest one was 10, 11 micron to 19 sometimes. So that's not good enough resolution. But I was hoping that some of these signatures would be preserved. Whatever is preserved on the micro scale would potentially be preserved on a slightly larger scale, such as, what's preserved on a 2 micron scale might be preserved on a 10 to 15 micron scale as well. But we don't know yet. We'll find out if the synchrotron mapping gives us any better results in terms of the clustering, etc. But better resolution in the synchrotron, absolutely better than the laser.
Great. So I think the next question was from Elva Escobar, who asked, have you analyzed any polymetallic nodules?
I have analyzed nodules, but I don't think they were polymetallic, no. No, I don't think so. But I've analyzed other nodules that weren't necessarily related to any kind of mineralization, economic mineralization.
So I saw that Elva also asked if you think the speciation is going to change with oxygen levels in the ocean.
Yeah, of course, the speciation can change with oxygen conditions, absolutely. But my point is that when you are in a controlled environment, in fact, there are some really good articles on molybdenum speciation. So there's different speciation even within reducing conditions. So I think in case of pyrite, because it's a microenvironment, I mean, it has to be reducing if you're precipitating pyrite. But even within that, you could see different species of, especially elements that have, sort of a gradation of species depending on the redox conditions. So, like I said, when you are orthogenically mineralizing an organism by pyrite, you need to have a very strong microenvironment control, because you can't let too much pyrite precipitate, otherwise you don't preserve much. So in that controlled environment, you could see difference in speciation, because certain organisms will have acquired different species for different functions, or just the microbial reactions that's causing the precipitation of pyrite might incorporate certain species of trace elements over the others. But that's still occurring in the context of a reducing condition. But would the speciation be different outside of the environment of the 100%? But I'm more concerned with what's happening within. And if you see, I actually don't know what I'm going to find. I'm very keen to see whether we will see homogeneity in speciation. We could even see heterogeneity. We don't know. So that's the big question. And I think one of the reviews of my proposal was like, well, what do you expect? And I was like, well, I don't know. I haven't done it yet. I don't know what to expect. That's the whole point of doing this this proposal or this experiment, to see if you see any difference in speciation and if they are homogeneous or heterogeneous, because you never know with biology. They're capable of both. So really keen to see what comes out of it.
Great. I saw that Paul also asked what the age of the fossils you were analyzing were.
I think they're Devonian, the ammonites at least, but my other fossils are Proterozoic. The one from the MacArthur Basin are Proterozoic. The ones from the Georgina Basin are Cambrian. So they're kind of all over the place. What do you mean by, sorry, I just saw a question. What do you mean by matrix? Yes, I apologize. So this is matrix, basically. So when I'm looking at using an in situ technique, you're targeting a specific mineral. So I'm targeting, for instance, pyrite. So anything on the outside of this mineral is the matrix material. It's the rest of the rock, basically, except the pyrite.
So the next question was from Jan or Jan, and I think it's might be a hard question to answer, but is there a specific kind of signature that can unquestionably be attributed to biological origins?
Well, yeah. So like the presence of carbon is considered to be pretty cool. And you can be pretty sure that it's biological, but I don't know. I have mixed feelings about carbon. I mean, you can easily get rid of it, especially given these rocks are, I mean, come on, they're like billions of years old. If it's not preserved, doesn't mean that, evidence of absence is not absence of evidence. So yeah, I think gosh, this is a very tough one. This is basically what I'm trying to answer as part of this thesis. But you know what? I think there is a signature. There is 100% a signature. We just haven't found it. There has to be something that's there. Like I said, I mean, I'm sure we can distinguish, or there is a way to distinguish between biological organization and mineralogical ordering. I most certainly have not found out the answer, but I think there is an answer. But yeah, morphology or texture alone is not going to tell us. Composition alone is not going to tell us. So yeah, it's very puzzling. But I hope, Jan, you're referring to textures. I mean, if it's a fossil, obviously it's a no-brainer, right? But like, it's textures like this, the one on the slide right now. I don't know what they are. But the fact that the pyrite is sedimentary, and it's microbial sulfate reduction. So in that sense, you could say it's biological. The precipitation itself is biological, but is it replacing, or is it orthogenically mineralizing this structure here, which was originally a biological organism? Now that's hard to tell. But did biology have anything to do with the precipitation of this structure here? Yeah, of course. I mean, microbial sulfate reduction. But that's about it. That's my limit of being absolutely sure. But I'm sure there is a chemical signature, but maybe one day one of us is going to find it.
So I saw a couple questions that are kind of related. And they're asking, can you think of anything that biologists or microbiologists can do to help kind of encourage collaboration in this kind of work?
Absolutely. Absolutely. I actually had I don't know if I'm sure you've heard of him, but he's a biologist in Italy. His name is Donato Giovanelli. And he gave a presentation on 10 things that Earth scientists should know about biology. And I was like, it was such an incredible presentation. And I think he basically said the same thing, that I think we need to work together, the geologists and biologists. I think we kind of do in the field of astrobiology, don't we? We try and that's the purpose of this seminar series, I hear. But absolutely, what we have in mind to test our hypothesis, for instance, growing, replacing or pyritizing some plant fossils in the lab and then testing it out. Of course, I don't have the experimental expertise that biologists would have. So absolutely, biologists would help, and I'm trying to collaborate with a few. But yeah, if you want to collaborate, please get in touch, or let me know. I'll get in touch with you. But it's absolutely worth it. There's this geology alone, or no, in actual fact, no branch of science alone is going to solve and answer these big questions, really. So absolutely, biologists can help.
Great. Yeah, I think that's definitely one of the goals of PCE3 is to kind of have this kind of collaboration with people from different fields. So that'd be great. And I'll have to look up that presentation you mentioned.
Yeah, it's really good. It's incredible. Very informative. I'm going to put my email address on there because I think someone asked.
Yeah, and I think someone also asked if you could.
Yeah, that's the guy I was talking about.
Hmm.
Oh, yes, sorry.
Great.
Yeah.
Yeah, I'll have to note that as well. I think.
Yeah, it's incredible. I think the scope for, I know we like to work in our own little departments of geology and biology and chemistry, but nature doesn't have these little departments. So if we behave more like nature, I guess we would, go far, get far. So yeah, that's the idea.
Great. I think I saw another question in there. think Richard Gordon asked if you think you can distinguish between archaea and bacteria.
My God, what? No way. My goodness, no way. Like for now, we can't even tell if it's biological or not, let alone archaea and bacteria. But maybe one day there will come a day. I mean, look, this is, if you're talking about archaea and bacteria in like modern sediments, I suppose you can. There's lots of genomic analysis and all kinds of stuff. But their actual preservation in the rock, especially like if they've been completely remineralized, I think that would be very tough. I don't know if, to my knowledge, I don't think there exists, given the size of these things. Yeah, it would be incredibly difficult. Maybe first we'll have to get through, differentiating between prokaryotes and eukaryotes first. And then maybe, I don't know, I'm not going to live to see that, I don't think. Maybe I'm being very pessimistic, but no, that's, yeah.
All
Big
right.
question.
I think that might have covered all of the questions that are in the chat as of now. So if anyone has some last minute questions or wants to raise their hand, go ahead and do that. And if not, I'll go ahead and thank Dr. Indrani Mukherjee for sharing with us today. And also thank you to everyone in the audience for joining. And if that's it for questions, we should be back in another 3 weeks for another seminar. And yeah, thank you again to everybody. All right, bye.
Thank you.